And now it gives me great pleasure to introduce Mr. Carl Zimmer, author and New York Times columnist. Carl will take us on a fascinating journey through the defining mystery of heredity, a journey through our genetic sense of belonging in the world, a journey into our past and our future. Carl will open Bloom with a keynote lecture on where we come from and where we will go. Please welcome to summer, Mr. Carl Zimmer. Carl, summer is yours.
Thank you. Thank you. Thank you, Svante. Thank you all for coming out. It's a pleasure to see you all, and it's lovely to be here in Copenhagen. I'm going to talk today to kick things off about heredity, about its past and its future. You might not think of heredity having a future. You might think that heredity is something timeless, but that's not true. Heredity changes. It has changed in the past, it's going to change in the future, and we play a part in that change. When I talk about heredity, what I generally mean is what it is that one generation passes down to the next. And there can be a lot of different things, but I'm going to focus today on one form of heredity, genetic heredity. And to get a sense of what I mean when I talk about heredity changing, just consider a piece of corn, an ear of corn. If you were to take these kernels and plant a new corn stalk, you would get corn that looks just like that. So we think of that as heredity, that the new corn plant inherits what the parent corn plant had. But corn has not always been corn. If you were to go back on Earth 10,000 years ago and look for something like this, you wouldn't find it. It didn't exist. 10,000 years ago, if you went to Mexico, there was a little weed, which we now call teosinte, and it produced little flowers and seeds. Didn't really look like much at all. But archaeologists can actually, let's see if I can point. Archaeologists can go back and find these early teosinte, which over time got larger and larger until they became the corn plants that we're familiar with. And it isn't just the size of the ear that changed, but it's the shape of the plant itself, the way that the corn cobs branch off of the plant. That has all changed, and we humans changed it. For many other plants, this is true as well. So once upon a time, if you wanted to find a tomato, this is what you would find in the wild tomato plant. And then this is what we have now. Obviously, this is not to scale, but relative to each other, you can see that wild tomatoes were tiny and did not have the same taste as the kind that we regularly use today. This transformation has also led to wolves, giving rise to new kinds of animals. And not just one domesticated animal, but a variety. These two breeds of dogs, they all descend from wolves. And there are particular changes to their genes that produce these different sizes. This is not a matter of, what kind of food you give these dogs. This little dog and this Great Dane, they will become those sizes because of how we have bred them. The irony is that for thousands of years we have done this, and we really haven't known what we are doing. And if you go back and look, for example, in classic writings from Rome or Greece, you won't find anything about how to breed animals or crops, or why crops are different than wild plants, because we just didn't really know. We had some ideas, but we really didn't know. Now, that began to change in the past few hundred years. There are some fascinating people who started us down the path to understanding how we are changing heredity. And by understanding it, we could have a better control over it. So, for example, take sheep. We have had domesticated sheep for thousands of years. We've had different breeds of sheep. But for the most part, people kind of imagined that really the way to breed a certain kind of sheep with a certain kind of wool would be simply to raise them in a certain way. So merino sheep in Spain, they were taken on these long journeys through the year into the mountains, to the lowlands, and it was believed that that journey gave them the right kind of wool, that they were a product of their environment. But in the 1700s, there started to become a new idea, an idea that maybe there was something inherent in the sheep that could be controlled and used to produce new breeds of sheep. And Robert Bakewell was an English farmer, and he had this idea in the 1700s that he would simply set out and make a new kind of sheep. And it was quite mysterious. He would pick out sheep from all around England and bring them to his farm. And he had barns that have no windows. And he would breed them in there in this mysterious way. And people thought he was some sort of wizard. And then he produced an entirely new breed of sheep called New Leicester, which turned out to be incredibly popular. And people would, pay huge amounts to buy one of the lambs of this new breed. And he was actually one person who wrote about him at the time said that they called Robert Bakewell the man who invented sheep, which is quite a compliment, even though because obviously sheep had been around for thousands of years. But he was the first person to really say, I'm going to make a new kind of sheep by somehow manipulating heredity. But still, Bakewell himself couldn't tell you exactly what was happening to the sheep. He didn't know. He had these certain rules about how to breed individual sheep, and he knew that if he followed these rules, he could produce a stable line of sheep that was different than other sheep. And every generation would stay the same that way. So it was a new kind of heredity. In the 1800s, people who followed Bakewell tried to do more of these experiments, more rigorous experiments. So Gregor Mendel was actually a big admirer of Robert Bakewell, and he wanted to use that approach to understand the rules of heredity for plants. And so he would breed different varieties of pea plants, and he discovered these patterns that we all learn in school. So, for example, if you were to take a pea plant that had a purple flower, was actually had these two different genes, and you bred them together, you would get this 3-to-1 ratio. That's because these are dominant and recessive genes. He didn't know these were genes. He could just see the patterns of the flowers. And so that was a profound discovery, but unfortunately, he didn't know where to go from there. He tried to replicate these experiments with different plants that have a very weird kind of heredity. He couldn't get these patterns again, and so he gave up. And other people just looked at his results and said, that's weird, and that was the end of it. So it was only after he died that the science of genetics was born, and Mendel was rediscovered. So what we know now is that those genes that Mendel was studying, looking at the colors of those flowers, are encoded on this wonderful molecule, on DNA. And so DNA has this classic double helix shape. And if you were to take all the DNA in one of your cells, it would measure about 2 meters, so from the floor to here, in every single cell. And It's basically, you see these are letters. There are these chemical units that DNA is made up of that are like an alphabet of 4 letters called bases. And then they're coiled up into chromosomes, which are then packed into a cell. Now, if you were to print out those letters I was telling you about on that 6 feet of DNA, those 2 meters of DNA, if you were to print it out and make books, this is what it would look like. Actually, this individual actually made books with a human genome in it. So you can see you would fill a wall in a library just with all the DNA from one human genome. And there you have thousands of genes that are encoding proteins and other molecules that make up your body. And other species have their own genome as well. And so that mystery that we saw before, how was it that corn became corn, is really a story about changing the corn genome, rewriting it. So you have mutations that arise naturally in corn and other things, and then if you cross them together, you can combine these mutations into new combinations. And then if there's some corn that is particularly good and you decide to choose that to breed, those mutations are going to become more common in the next generation. And over enough time, those mutations are in all the corn. So scientists now can actually go back and look at the corn genome. These are chromosomes of corn. And they can compare the corn and the corn's DNA to the wild ancestor, the teosinte, because teosinte is still growing. And you can see the key places where they're different. And so this graph shows you basically those peaks are places in the DNA where we have changed the DNA to an extreme. So to turn teosinte into corn thousands of years ago involved us changing hundreds and hundreds of different genes. And these are genes that changed the shape of the ear of corn, made it big. It changed the way that the kernels are produced. It changed the shape of the corn plant itself. But things didn't stop there, because corn began in the lowlands of Mexico, and then it spread. So even before Europeans arrived, discovered corn being grazed by Mexicans and spread it all over the world, corn was spreading to other parts of the Americas. And as it was spreading, there were little adjustments that were being made. Farmers were making them, not realizing it, as they were breeding them for growing in mountains and growing at different latitudes. There's a different amount of sunlight in Canada than there is in Mexico. And so actually, it turns out that there are certain mutations that are like dials. You can dial up a certain protein or dial up the time when a plant will flower. And that's an adaptation to grow corn in more light or less light. And now we can actually see these kinds of changes. So we're able to look back at the past now and start to see how we have genetically altered plants, animals, how we ourselves have genetically changed over the past 50,000 years. And with that knowledge, there is the potential to make those sorts of changes for future generations. Now, originally, our ability to control heredity beyond just breeding plants, for example, was fairly crude, but it's led to some important successes. So, for example, in Hawaii, the papaya is very important. It's a very important crop. And there was a virus that showed up on the island and began to devastate the crops. And this was really putting the whole industry into danger. It turns out that that a protein from the virus itself, if it's in the plant cells, actually makes it harder for new viruses to infect them. It's a very counterintuitive thing, but basically this virus protein will interfere with other viruses. So what scientists decided to do is they said, well, imagine that this is that blue bar is the papaya genome. Let's take the gene for that protein, and let's just try to insert it into the papaya DNA. So now the papaya will make this protein by themselves. So this is a plant virus, and so this protein is completely harmless to people. I mean, you actually, when you eat fruits and vegetables, you're eating plant viruses all the time. But the thing is, they're adapted to the plants, not to you, so you just grind them up in your stomach. That's not an issue. But papayas, if they can make this protein, they are resistant to the virus. And so this change, this relatively simple, crude change, actually saved the papaya industry in Hawaii. But this is a very early, simple kind of genetic engineering, where you take a gene, a whole gene, from one species, and you try to put it into another species. But scientists have long wondered, well, could we do something more sophisticated? Could we, rather than grabbing a whole existing gene and moving it somewhere else, what if we had the ability to rewrite DNA? We'd need to know what we wanted to rewrite it into, obviously, and we'd need the technology to do it. But if we could rewrite DNA, then we could make much more subtle, much more sophisticated changes. And this could have a really powerful impact on things like our food supply, on medicine, all sorts of things. It would be a potentially very valuable tool. So this is the background to maybe one of the most important inventions of the 21st century. I mean, obviously, we're only 19 years into the century, but I'm going to make a bet that when people look back, what happened in the 21st century that really changed the game, I would argue that CRISPR will be one of those. Now, you might say, well, what are you talking about? What is CRISPR? It's a very weird name. And you might think it's some sort of food product. It's an acronym, and unfortunately, scientists have a way of thinking that acronyms are useful. If you want to know what CRISPR is, this is what CRISPR is: clustered, regularly interspaced, short palindromic repeats. Commit that to memory. You'll impress all your friends. I'm not even going to tell you why. I'm not going to even tell you what that all means at the bottom. We need to move on. But the fact is that scientists discovered in bacteria that bacteria have this extraordinary capacity to fight off their own viruses by making molecules that could zero in on the virus genes after they invade them and cut them up. What this meant was that the bacteria could make molecules that could recognize DNA, could recognize specific bits of DNA, specific strings of DNA. Because obviously if you're a microbe, you don't want to make molecules that are going to destroy your own DNA. So you need a very sophisticated way of recognizing invaders, recognizing yourself. And bacteria can do this. Bacteria are amazing, but we didn't know that bacteria could do this until about maybe 10 years ago. And what scientists realized was we could take this natural technology, this thing that microbes have done for billions of years, and we could adapt this and make it a tool, our own kind of tool. So the basic idea of CRISPR is that you can actually engineer a molecule that will be able to find one particular bit of DNA. So in this diagram, this thing called sgRNA is actually, it has a structure that will let it zero in on one particular place in your DNA. So to think of all those books, imagine that you could just take a molecule and throw it at that set of books, and it could find the right page, and on the right page, it could find the right paragraph, and on the right paragraph, it would hit the right letter in the right sentence and cut it, blot it out. This sgRNA actually brings with it sort of molecular scissors. So what happens is that you find this site that you want to cut, and these scissors make the cut. So what you can do is you can say, like, I want to get rid of this one letter of DNA, or maybe 10 letters of DNA. Now, if you just get rid of it, then the DNA will actually repair itself, and it'll just be missing that section that you cut out. Or you can drop in another piece of DNA to put in its place. And in some experiments, it's possible to change just a few letters of DNA. Sometimes you can change just one letter. And this actually now is working incredibly well in terms of the accuracy, in terms of the speed, in terms of the cost. Scientists are using CRISPR all the time now. These 4 scientists, Jennifer Doudna, Emmanuel Charpentier, George Church, Feng Zhang, were 4 among a number of scientists who recognized what bacteria were doing and pioneered how to use this bacterial trick as a new kind of gene editing tool. And so if you go to universities around here in Denmark or elsewhere in Europe, you will find scientists using CRISPR all the time. If scientists want to do an experiment to understand how a gene works, you can just use CRISPR, say, in a dish of yeast or in an experimental mouse to change the gene and then see how the organism changes as a result. So one of the things that you can do is that you can change the structure of a protein. So here's an example where you've got this, it's the same protein, and one mutation can change one little part of it. Might not look like much, but actually, in this particular case, if you have the blue version of this protein, you are at much higher risk of developing ALS, also known as Lou Gehrig's disease, a devastating neurological disorder, what Stephen Hawking died of. So little changes can have big impacts. In addition, you can also change pieces of DNA so that you're not actually changing the protein, but you're changing the dial I told you about. So if you wanted a cell to make more of a protein, you change the dial using CRISPR. So what are people using CRISPR for now? One of the most interesting places that they're applying it is, again, to plants. So instead of the old-fashioned breeding, instead of moving a gene from one species to another, researchers are wondering, well, if we just make a few tiny changes to the DNA of a plant, can we produce a new kind of plant that's valuable to us? So one example I talk about in my book involves a plant called a ground cherry. It's a wild plant, and it produces, this is the cycle from the flower and then to the fruit that it produces. And the flower turns into kind of a paper sack, and inside grows this fruit. It's delicious. If you ever get a chance to eat a ground cherry, eat it. But the problem is that it's not very good as a crop. It's hard to raise in large numbers. It's hard to collect. The fruits fall to the ground and they will rot quickly. These are the challenges that early farmers faced with things like teosinte. They turned those wild plants into crops that were much easier to harvest. And so a scientist named Zachary Lippman at Cold Spring Harbor Laboratory in New York has actually been using CRISPR and saying, well, if I just change a few of these bits of DNA, I can change the dials. so that you can change the timing at which these plants grow. And his goal is basically, with maybe 5 or 6 tiny changes to individual letters, he hopes that he can turn a wild plant in 1 generation into a domesticated crop. So something that would have taken centuries before could potentially be done in a matter of months. So that's the kind of power that CRISPR may potentially give us. There are more ambitious ideas, more controversial ideas about how to engineer the natural world. No one has actually done these things yet in the wild, but there have been lots of experiments, which I write about in the book, about what would happen if you start to using CRISPR to engineer wild animals. Why would you want to do this? Well, one wild animal is the mosquito that spreads malaria. So the parasite Plasmodium, which is infecting those blood cells there, it's carried inside these mosquitoes, and then when they bite someone, they can spread the disease malaria. Several 100,000 people a year die of malaria. We have some medicines that work okay. We have no effective vaccine yet. And so some people have argued that we might be able to use CRISPR to alter mosquitoes as a way of eradicating malaria. And it's really fascinating how this would work. And so I'll show you how it might work. So scientists have found that they can alter a gene in mosquitoes that will make them resistant to the parasite. The mosquitoes themselves cannot harbor the malaria parasite if you alter this gene. They make an antibody that kills it. Now you could say, well, okay, just make a regular engineered mosquito with that gene, and then it'll release it. And what would happen? Well, probably what would happen is that engineered gene would disappear from the gene pool. And that just brings us back to good old Mendel. So what Mendel realized was that, in effect, what he realized was that you have 2 copies of each gene, and so there's a 50-50 chance that each offspring will inherit one of those copies. And so if you have this orange box here representing that engineered gene, and you just release that mosquito out into the wild, chances are, since all the other mosquitoes don't have it, it's going to become rare in the population, and it might just disappear after a while. It's pointless. There's really no point in engineering 1 mosquito or a couple hundred mosquitoes and setting them loose. It's not going to help you. So here's where CRISPR gets a little weird. Imagine that you actually put the genes for CRISPR into a mosquito so that it can use CRISPR on itself. Now, I know this is a little strange, but bear with me. So you start with a regular gene. You change it now so that you have this gene for resisting malaria, and you also have genes for the molecules of CRISPR. So what will happen is the mosquito will make its own CRISPR molecules, and they actually go to the other copy of its own gene and rewrite it. So it's rewriting its own DNA, cutting out the old gene, putting in the new one. So now this mosquito that started out with 1 copy of resistance to malaria now has 2. What happens if you release these mosquitoes into the wild? No one has done this yet. No one has done this yet. I've gone to labs where they've created these mosquitoes. It works in a lab. No one's going to release these mosquitoes until we're absolutely sure that this is a good idea. But you would go from this 50% rule I was telling you about. With CRISPR, it would be 100%. You would break Mendel's law so that you would release a few hundred mosquitoes into an area, and everything looks like, if you do that, the possibility is that really within a few years, all the mosquitoes in the region would be resistant to malaria. They couldn't carry malaria, therefore the parasite would be eradicated. So this is a really profound possibility. Scientists are working on it, but no one is going to be releasing these things anytime soon. There's a lot of debate we have to have about whether this is a smart idea or not. What role do mosquitoes play in ecosystems? Are we going to disrupt that in the process? But I would also argue that not doing anything about malaria is an ethical choice, just as experimenting with new treatments to affect malaria. So yes, there are risks here that we need to evaluate, but to simply say like, oh, I don't feel comfortable with this and therefore I'm okay with several 100,000 people a year dying, that's not acceptable. Maybe there are better ways of dealing with malaria, vaccines, for example, other public health measures, bed nets and so on, but we can't just say that doing nothing is okay. It isn't. So one possibility is that CRISPR, with this kind of approach, it's called gene drive, might be one tool that we might use. So I've talked about using CRISPR on plants to change heredity. I've talked about changing mosquitoes. What about ourselves? That is one of the most controversial issues with CRISPR right now. But we've known this has been coming for a while, actually. In my book, I write about a American microbiologist named Roland Hotchkiss, and in the early 1960s, just from the research that had been going up till then, this is over 50 years ago, he saw exactly what we would be dealing with now. The possibility of using engineering to alter the future of our heredity, to change our genes in ways that would pass down to future generations. And he said, like, this is going to be a profound thing, but he was very confident that people were going to use this power as soon as they invented it. He said, we are going to yield to the opportunity. We are going to yield when the opportunity presents itself. And so over 50 years since he said those words, the opportunity has presented itself. So I'll give you an example of how scientists want to use CRISPR to cure fatal diseases. The case I want to give you is muscular dystrophy. So Tyler Zielinski is a boy who had inherited muscular dystrophy. What happens is that it turns your muscles to jelly. People with muscular dystrophy usually die in their teens. Sometimes they manage to live into their 20s, and then they die. Their hearts give out, their diaphragm can no longer function because their muscles aren't working. There is no treatment, no cure, nothing. And the irony is that a lot of diseases are kind of mysterious. We're still not really sure what causes them, or maybe one disease is 100 different diseases. This kind of muscular dystrophy, we understand it down to the molecules. If you look at a muscle fiber, there is a protein called dystrophin. Muscular dystrophy is just from a mutation to the gene for dystrophin. Normally, dystrophin makes the force that pulls in a muscle. These proteins don't work because of a mutation. So if you can rewrite the gene for dystrophin, then you could treat the disease. It's an entirely hereditary disease. It's carried on the X chromosome. If you remember, women have 2 X's, men have 1. A woman can carry the mutation and be healthy because she has a working dystrophin gene. But a boy, her son, might inherit the 1 X copy of the X chromosome that has the faulty gene. And if he inherits it, he gets muscular dystrophy. It's quite simple. So what a group of scientists have been doing is they've said, what if we could create CRISPR molecules, put them in a virus that only affects muscle cells, inject the virus into people with muscular dystrophy. The virus would infect the muscles, and the CRISPR molecules would rewrite that gene. They'd make one change to one part of the dystrophin gene. It would repair this genetic defect. So what would happen? So Eric Olson is a scientist in Texas, UT Southwestern Medical Center. And he has been testing this possibility just in the past couple of years. This is very new research, and it's very promising. He's been working on this in mice and in dogs. These are animals that have the same mutation as people do who have muscular dystrophy. Their muscles have the same defect. So in ordinary muscle taken from an ordinary dog, if you give it a stain so you can see dystrophin, this is what it looks like. So that's what you want. Now, a dog with, there we go. A dog with muscular dystrophy, if you look at their muscle, that's what you see. You don't see any dystrophin because the dystrophin the normal version of dystrophin just isn't there because of the mutation. And if you give them these viruses I told you about that carry CRISPR to their muscles, this is what you see. These same muscles have had their DNA rewritten. They are now making dystrophin again. And in fact, with studies on mice, Olson has actually shown that they regain some of their muscle force. So within I would predict within maybe 4 or 5 years, there will be clinical trials on people using this technology, using CRISPR to cure muscular dystrophy. We don't know if we never know if these things will work until they're tried out. But I think we have good reason from experiments like this to think that they will. So this is the kind of really revolutionary kind of change that you can expect from understanding our heredity and how to manipulate it. Now, if changing people's genes in their muscles obviously doesn't change the genes that they pass down to the next generation. But CRISPR does have the potential to do that too, if we so choose. So think about this. Think about the fact that what I've just described to you would involve diagnosing someone with muscular dystrophy, maybe when they're 5 years old, 10 years old, 15 years old, after they've already had years and years of suffering with this disease. One could argue that, well, maybe we should just fix this from the start. What do we mean by the start? Well, you could potentially use CRISPR on fertilized embryos. In fact, scientists have already worked out how you can use CRISPR on fertilized human embryos. Scientists know how to do this. So the possibility arises, the theoretical possibility, that you could use CRISPR to change the DNA in embryos to get rid of these mutations that cause these sort of diseases. Muscular dystrophy, sickle cell anemia. There are thousands of these diseases that you could potentially use CRISPR on human embryos. But remember, that's an embryo. So that person who would develop from that embryo, if they had children, they would pass on that change to the next generation. So this is a profound moment when we can possibly do this. And the ethics of it are not simple. Some people might say, well, who could argue against curing muscular dystrophy? We should just be doing this right away. We should just put this into action as soon as possible. But, then there are other things that you could imagine doing, things that seem like they have a reasonable medical basis. So, for example, there's a gene called Klotho. And there's a variant of Klotho, a naturally occurring variant, there probably in about 20% of you, that gives you extra protection, it seems, against Alzheimer's disease. It makes your brain more resilient. So, let's say that you're going to use in vitro fertilization to have children, and your doctor says, what do you say, while we're at it, do we give your child this variant? I mean, you want to give your child every protection against Alzheimer's disease when he or she gets old, right? Right? I mean, what kind of a parent are you? Now, the tricky thing with Clotho is you might say to yourself, yes, of course, I want the best for my child. I want to protect my child as much as possible from diseases they might encounter in their lifetime, whether it's muscular dystrophy as a child or Alzheimer's disease as an adult. Sure, what could be simpler than that? Well, Clotho has a little twist to it. The same variant I told you about that protects 20% of you from Alzheimer's is actually making your brains right now run a little better. It improves learning, it improves memory. So what is this? Is this protecting you from a disease, or is this enhancing you? And when we start talking about using genetics to enhance children, and these enhancements can be passed down to the next generation, this is when things get, a little scary. Like, what are we doing? Can we trust ourselves with this kind of power? Should we just let individual parents make the decision? Does that mean that we just let the parents who can afford to make the decision, and everybody else has to just deal with the genetic weaknesses that we're all prone to? Are we going to create sort of two populations, a rich and poor, where the differences are baked in genetically? If you don't like that, well, okay, if you have a universal healthcare system, CRISPR for everybody. Do you feel okay with that? Or are we going to be getting rid of the genetic diversity that is so important to ourselves as a species? Now, these are the sorts of questions that really until maybe last year, you could just sort of talk about late at night, preferably over a lot of beer. But it all seemed hypothetical, and it really is not anymore. So in November, the world discovered that a scientist named He Jiankui in China had gone ahead and done this. He had found at least two couples to become subjects of a CRISPR experiment. And what he did was he took these embryos from these parents and he engineered one gene in them. The claim was that these babies would then become resistant in life to HIV. He made that change. He implanted the embryos with the help of fertility doctors in the women. And in October, twin girls were born with this change to them. There's another woman who supposedly is still pregnant with another CRISPR baby. We'll wait to see what happens there. But we've already entered into the age of CRISPR babies. We're here now. And unfortunately, this was probably the worst way to enter this period. This was an incredibly bad, unethical experiment. The informed consent was not done well. He Jiankui was not honest with the hospitals and the funding agencies. No one really knew what he was doing until he told the world, until these babies were already on Earth. That is not how we should be thinking about CRISPR. We should not be blundering into this without wrestling with these complicated questions about heredity. And we need to do better. We need to do better because CRISPR is just the start. And just before I finish up, I just wanted to talk about a different way of editing DNA. It's actually not editing DNA, it's writing it. This is an article I wrote last week in the New York Times. What scientists did was they took the genome of the bacteria E. coli, it's about 4 million letters, And they decided they wanted to make a sort of system-wide change to the way these bacteria work. This would involve about 18,000 changes. And you can't do that with CRISPR. With CRISPR, you could change one letter or one little chunk of DNA. That's about all you could do right now with it. So what these scientists did was they basically, like literally on their computer, like imagining you're writing a story or a cookbook for E. coli, and you just go in and you do sort of a global search and replace 18,000 different times. So now you have the text on your computer. What these scientists then did was they made the genome. In other words, they actually produced the physical molecule of DNA that is that new E. coli genome that has never existed before. They made it. They built it in pieces. They put the pieces together. And then they just said, like, all right, let's see what these bacteria do with their new genome. Do they boot up? And they did. They grow. They're fine. They're a little funky. They're like, the E. coli are shaped like a rod. These are a bit longer. No one knows why. But they're alive. They live. So this is genome synthesis. And it's going to make CRISPR look like child's play, because we are going to be able to write entire genomes from scratch. I don't think we've quite come to terms with what that will do to our own sense of the past and the future, of heredity itself. And we have to come to terms with these issues because the technology is moving ahead so fast. These are exciting times, these are frightening times, but hopefully we can put these tools to good use in the future. So thank you so much. I really appreciate you spending the time with me today, and I hope you enjoy the rest of the festival.